thanks to everyone of you to be here with us tonight. It's a first. It's beyond disciplines. It's a very exciting moment because when we thought about this, we thought about creating a place where people could meet and where people can talk and discuss. And one of the things that we thought is to organize the first one at Loyola. And when we said that, people say, people won't come. And I think those people were wrong. So thank you so much. It's a full house, and we're very pleased to have you here. Now, beyond disciplines. Beyond disciplines is to illustrate the diversity of the Faculty of Arts and Science. It's to illustrate the vibrancy of its intellectual environment. And it's to illustrate how meaningful conversations can happen from a whole set of viewing angles. And you'll see it tonight. We are a very diverse faculty from philosophy to biology, as being presented here, to political science. And some people ask, how does it hang together? It's not just because there's a faculty. It's because we have great people that can actually exchange on meaningful topics. And the one of tonight is very meaningful. The other thing about it is you might ask about the name, Beyond Disciplines. I had the privilege at one time to be at a quite well-renowned university in the UK, and they had a series called Tipping Point. I thought, this Tipping Point, this is a great series, Tipping Point. And another series was called Across the Divide, and we discussed that internally. Across the Divide. Yeah, but are we so divided? We're not that divided. So where's the divide, right? So we thought about provocative thoughts, and it's right there. And we said, well, you know, maybe that's not quite it. And then we commissioned our communication advisor with some, you know, we sort of discussed these names. We talked about disciplines, and then undisciplined came up. And we said, maybe not. So Beyond Discipline is what it is. And we're very proud to have its inaugural event tonight. Our faculty is a big federation, and I welcome all of our staff, students, and faculty members and chairs to this. But also, this event is open to the outside world. So it's a public thing. We own it as the Faculty of Arts and Science at Concordia, but it's a public event for us to expose knowledge and questions to everyone. So welcome. Tonight's topic is emerging as a critical issue in our society, and it clearly spans the sciences, politics, and philosophy. And luckily, at Concordia, in our faculty, we do have experts that are actually connected meaningfully with the topic and coming from all these point of views. So welcome to our panelists. And this is, we thought, a perfect theme, really, to get us started. As a special treat, after the event itself, there will be a visit of Vince Martin's lab. For those who are interested in how the science is taking place, Vince is as one of the sort of cutting-edge lab in the field. And so there's a sheet of paper that will circulate, so you just sign up if you want to visit a lab. And it's a fantastic experience, I'm telling you, so as a selling point. So the sheet will circulate as... As all of you, I'm very eager to get this going, so I'll stop now. And I'll introduce David Secko, who will be the moderator. David is from the Department of Journalism, and he will act as the host and moderator of this evening. So enjoy it. It's going to be great.
Thank you, Andre. Thank you, everyone. Welcome tonight. I'm going to go quick because I think we have a lot to discuss and hopefully a lot of questions that are brewing in your mind. The one thing I'll say is that since the idea here is beyond disciplines, kind of expanding your mind and thinking that way as we go is a good way to start this in terms of thinking of the questions you might come up for the discussion in that way. As was mentioned, I'm a associate professor of journalism in the department. I'm also a former science journalist, and I did some PhD work in microbiology. Actually, Vince was leaving as I was coming in the same laboratory setting, the same building, in that kind of a sense. And back then, I really could feel, basically, in all the graduate students I was working up with, this pent-up desire, this strong pent-up desire for a genome editing tool that will allow them to do things quicker, more precise, and more like lively than they had at the time. It was everywhere. We did a little bit of editing with genes and things like that, but nothing like what CRISPR would allow now. And when people see it, when I see my colleagues from those days and their eyes light up, when they talk about this topic, I thought it was a fantastic one to start with. So you may have heard about CRISPR in the news, you may have heard about the spin, the hype, all the stories about sexism and how it works, how it might go back, or you might not know about those stories, but there's lots here for us to untangle. So I'm gonna, since it's a big topic and there's lots to talk about, I'm gonna hand it over to our first speaker to get us off, so let's get started. What I'm gonna do is I'm gonna introduce each panelist as they go and give them their allotted time, and I will be over there kind of yelling and screaming if we have to go, and that's to you, Vince. And so we know you got your 5 minutes. All right, and we'll go from there. So the first one I'm going to introduce up is Alisa Piekny. I'm going to pronounce it right, an associate professor, close enough, in our Department of Biology. And I'm going to hand it over to her to introduce the topic to us and to kick off a great night. Alisa, thank you.
Okay, so I've been given the task to try to explain the science to you. So I'm going to try to do this as painlessly as possible, but if you have questions, of course, I'm happy to answer them. So first, I just wanted to bring up this, what's the big deal about CRISPR anyway? We have had genome editing tools available to us before. This is not the first one. But what I'd like to emphasize is this is the first one that's really cheap. It's also very easy to use, okay? So those are two very important things for those of us that need to do this kind of work with our research. And now this has obviously huge implications for ethically if it's going to be used for editing human genomes. But for those of us that work with model organisms, this has been really a great advancement. And really I'll just sort of highlight here, has really opened the door for editing the genomes of many organisms. But again, there's some obvious ethical implications when we now discuss the editing of human genomes. So what is CRISPR? Okay, it's actually a naturally occurring defense system that bacteria have to fight against infection. So bacteria can get infected just like we can. These are called phage. And these are these little these green things that are attached to this cell. And what they will do is inject their DNA, and then that DNA will make more phage, and then they burst out of the cell and kill the cell. Not a good thing, right? So what's amazing, though, is some bacteria, when they're exposed to this phage, resist it. They can actually survive the infection. And so this was obviously an area of research for people for many years. And then what they discovered is that their ability to successfully defend off this infection was because they had actually acquired a small piece of DNA from that phage and incorporated it into their own genomes. And this is, do we have a pointer?
Can somebody get a pointer? Have we forgot?
No. All right. I know. Oh, here we go. Here we go. I got a mouse. Okay, so this little, what happens is there's a little piece of DNA that gets cut from the phage DNA and gets incorporated into the genome of the bacteria. And these are called spacers, okay? And what happens is there are multiple of these spacers will accumulate in this genome with exposure to different DNAs. And these are actually sandwiched between these repeat regions, which are just small pieces of DNA that can form interesting structures, okay? And so because of this, this system has been given this really interesting name, right, which is an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, okay? Something else I'd like to point out is that there are these genes that are also part of the bacterial genome, and these genes make the workhorses, the enzymes that are then able to go and destroy this phage DNA, okay? So this is the important part that you need to know. And I'm going to skip over this for time. What I thought I'd do is just show you how a phage actually infects the bacterial cell, and then how this CRISPR system takes care of that, okay? So if you have a phage infecting a cell and you have DNA that's coming from that phage, then you'll have some enzymes will come in, and they'll be able to cleave that DNA, take a small piece, and incorporate it into this CRISPR array that I've already told you about. Now what happens is the next time that phage, the same phage infects that bacterial cell, now the bacteria has a way of recognizing that DNA, okay? Because these segments will get processed, and then they'll get incorporated into a module that contains these small little RNAs, and it's okay if you don't know what RNAs are. Okay, they're just small little segments that are perfect copies of those little DNAs that they had incorporated. And then they get incorporated with these enzymes, and then now they can go and attack that phage DNA that perfectly matched that sequence. Okay, so that's the science behind it. Now, how do we take this defense system that works in bacteria and now use it to edit genomes? So it turns out that people were able to clone these workhorse enzymes that actually do the job of cutting the DNA. And then they figured out a way to design a system where you could have these little matching sequences incorporated into these modules as well. And so this is built as a sort of all-in-one system. It's really easy for us to use in the lab. All we have to do is put in our sequences of interest here. We have a vector that makes the enzyme, and now we can just inject it into our cell or our organism. And what happens is this module will have the information it needs to go to your DNA at the specific site that you're interested in and now cut it. Okay, so we now have a way of cutting the DNA, getting to the site we want to cut. How do we now create mutations? Where's the editing part of this coming in? So this is a bit complicated, but I'll just briefly, hopefully, describe this to you. So there's two ways that DNA can repair itself when it's cut, okay? One is called non-homologous end joining. This is an imperfect way of repairing that cut, okay? So if you, for example, added this workhorse enzyme with this specific sequence to go to the site that you want it to go to cut, and that's all you do, then what will happen is the DNA will try to repair itself, but it won't do it very well. And what will happen is you'll end up with mutations at that site. The other type of way of repairing is called homologous recombination. And in this case, what we can do is add what we call a donor DNA. This is DNA that would have overlapping sequences that overlap at that site that we're trying to target, but then we can introduce anything we want. So let's say I introduce GFP, which is a green fluorescent protein, and now I can make a green fluorescing organism, okay? And this is just showing how this could work if we were to make, say, a heritable mutation. We can use this enzyme that I already told you about, the specific sequence, inject it into a zygote for a mouse, for example, and now the mouse will grow up with these mutations in them. And these are heritable, so they will pass this on to their next generations. And if we do the same type of thing, but in this case we have our donor template, now we will make mice that have this insert in them, okay? So now I just wanted to very briefly tell you who discovered CRISPR, okay? So now you have some sense of what it is, what it can do, what we're using it for in the lab. Now I just want to get it out there, there is no one discoverer of CRISPR, okay? And just like many things in science, these are work done by teams and teams of scientists, various leaders, people working in the labs. And really back in the 80s, they already had a sense that there were these arrays existing in the bacteria, okay? And it was really only in 2007 that there was this really seminal publication that really brought everything together. They figured out that there were these foreign pieces of DNA that were getting incorporated into the bacterial genome, and that this was being used as a defense system to then target and destroy foreign phage. Okay? Now, once we hit this 2007 mark, this is where we really now get into more modern use of CRISPR. And there are 3 scientists, and I initially was only going to talk about these 2 women who I think deserve due credit, but I decided to also bring in a local celebrity as well. So we have contributions from Sylvain Moineau, Emmanuelle Charpentier, Jennifer Doudna. What they were able to do is really start harnessing the power of CRISPR. Let's take this bacterial system, and now let's make it work for any genome. Let's make it into an editing tool. And so they did a lot of in vitro work to figure out how the enzymes work, how they come together with these guides, how they reach their targets. And around this time, after their discoveries, 2010, 2011, 2012, this really now exploded the community. Those of us that work on model organisms, such as C. elegans, zebrafish, frogs, mice, all immediately had a tool available to them that was very cheap and easy to use, and immediately started editing genomes for research purposes. And I'm just showing you some green organisms over here to give you a sense of some inserts that can be placed in a C. elegans mouse or a zebrafish. But I have to just briefly mention another person. This is Feng Zhang. I wanted to introduce him because he really also made some significant contributions in allowing the technology to really be translated into using complex genomes, so mammalian cells, mouse cells. He's also, of course, at the head of a controversy, which I think we'll hear more about later. Of course, he has filed many, many patents on trying to patent CRISPR and its use for genome editing. And of course, he's had some disputes with the ladies that I just presented to you earlier. We'll get into that later, I think. And I just also wanted to mention, right, so now we're moving beyond just these model organisms that researchers are using in the lab. And in 2014, CRISPR was used to now modify monkeys, primates, so these are very complex systems now, and you can obviously immediately grasp the implications of this work. So the first monkeys are born in 2014 that had been edited by CRISPR in exactly the same way that I already mentioned to you. And in 2015 in China, the first human embryo to be edited was published. So we now move into, I'm wrapping up right now. And so now I think this is a good segue into some of the other topics. But I just wanted to mention that Britain has now also given the go-ahead to edit human embryos. And since the discovery of CRISPR, we've had really an explosion in the community in terms of the number of publications, the patents that have been filed, and of course, the funding that has now been given toward CRISPR work. So I'm going to stop there. Thank you.
And I think these figures, which I'll leave up for the moment, are really telling of where this is going and how this is playing here. Thank you, Alicia, very much for a great introduction and a great talk on that. I'm going to ask our next speaker to come up right away. And our next up speaker is an undergraduate student also from the Department of Biology, Adithi Krishnan, who is doing her thesis project with Dr. Malcolm Whitney. And she's going to come up and talk a little bit about the use of CRISPR in the laboratory and her own experience.
Okay, which one is it?
Should be number 2.
Number 2, okay. Sweet. Okay. So, hi everybody. I'm an undergraduate student, like he mentioned. I work for Dr. Whiteway. And I'm just going to give you a very brief introduction or a brief talk about my project and how I use CRISPR in Candida albicans. So Candida albicans is a major human fungal pathogen. It has been shown to have lethal infections in immunocompromised individuals, which is why we study this species. Candida has pretty complex molecular genetics. So it's a diploid fungus, which means you need to knock out both alleles to see an effect. Also, it has poor homologous recombination. But researchers have still identified a CRISPR system to use in Candida. I'm not going to go into the details of this, but our lab uses two specific systems called a solo system and a duet system to induce genetic changes, to induce mutations in the Candida genome. These are just cassettes of the duet and the solo system that we use. So Dr. Piekny mentioned that there are several aspects to the CRISPR experiments. So there's a guide, there's a PAN sequence, there's several technical aspects. So whenever we conduct these experiments, there are several aspects that go into it to end up with our mutated DNA right here. A good application of this is a gene called the ADE2 gene. This is a gene found in yeast or Candida that I work with. So it's been shown that whenever there's a mutation in this gene, we see red colonies. So what we wanted to do was use CRISPR to induce this mutation and see how efficient it was. So as you can see here, so we used CRISPR to induce a mutation in the ADE2 gene, and we see several red colonies. So a very, very attractive aspect of CRISPR and Candida is that it has a very high efficiency, so high that we don't need to select for our mutated colonies. Also, it can target more than one loci, so the fact that it's diploid and it can target more than one loci is also very, very attractive. And we can observe loss of function mutations in essential genes, which is normally very hard to do. So how does this apply to my project? So the goal of my project is to look at the interactions between immune cells and Candida to see how immune cells react to Candida. So what I do is I take immune cells and I take Candida, I put them under the microscope, and then I look at it over time. So far what I've seen is that after 2 hours, the immune cells engulf the Candida. What I'd hope to do with CRISPR is to identify target genes that would alter this interaction between the immune cells and the Candida. So I'm trying to knock out one specific gene called MNN23, and I want to see if that will maybe make the Candida more resistant to being eaten by the immune cells or make them more sensitive to being eaten, essentially. So, yep, that's it.
We got other people controlling things now. Thank you very much, and thank you so much for the talk. And I love to hear in the lab experience of getting this done. I'm going to go right to our next speaker, Dr. Vince Martin, who's also a professor of biology and is running one of the labs that you might be able to see later. And hand it over to Vince to talk more about CRISPR.
Okay, so after all this technical stuff, I decided to dumb mine down quite a bit. So there's no gene names or anything like that, but there's lots of nice pictures. What I was tasked with is to kind of give you an idea. So personally, when people start talking to me about CRISPR, I get very frustrated because the only thing they talk about is editing genomes and human genomes mostly. And the tool can do so much more than that. We have a glimpse of what we can do with model organisms from Alisa, but once CRISPR got developed, people got pretty creative around the things that you can do. So I hijacked this from Alisa's presentation. This is what most people think of when they think of the CRISPR-Cas9. When I think of CRISPR-Cas9, I think of a protein and a little piece of nucleic acid, and it allows the two of them to home in on a very specific region of a large genome. That's all. If you're going to retain something from this talk or something from this entire event, very simple thing. You want to take this protein to this particular location, and that's the strength of that tool. So that's the strength of the tool for editing genomes, because you can find that very precise area in the genome that's very large. We have very, very large genomes. So when you're thinking about editing, Alisa showed this already. You can cut the genome. I wish I had a pointer. I wish I could CRISPR myself a pointer. So you can cut the genome and get errors, and you can cut the genomes and put things into it. That's very, very simple. That's some of the early stuff that people figured out how to do. And again, this is what was in the popular press. I'm going to skip that right away. As I said, think of this tool as a homing tool. And once you know what that can do, then you can start getting really creative on this. So the example I give here, so let's say you have the little piece of DNA that brings your protein at the right place. Let's say you put 1 2 of these guides together with the one protein. So what does that allow you to do? As you can see up here, you have target A, target B. So now instead of taking your DNA and opening it up, now you can take your DNA and cut a big piece out of it. Right? So all you've done now is add one extra guide to home in on a different region next to it. So now you can decide to cut a little piece, a big piece, or a huge piece. And then you can ask the question, what happens if I cut this piece of DNA that much, that much, that much? Right? So you can start asking new questions that you couldn't ask before simply by adding that little extra piece of DNA into that. So that's one thing that was pretty useful, and we're actually using that in the lab. Here's another tool that people started developing, which is pretty powerful. So here's another question you can ask. I have a cell, and I want to make a bunch of mutations into the cell, and then I want to ask the question, what did each of these mutations do? So now instead of using 2 of these little guides of RNA, or 2 of nucleic acids, you use, my kingdom for a pointer, you use what we call a library. So you'd use 10, 20, 50, 100. So all of a sudden your guides, it's a big library of guides, are targeting 50, 100 regions in your genome, and they're going to make mutations in there. So now all you have to do is dig through all of these mutations and find what you're looking for. So there's another tool. Make libraries, large libraries of mutations that are very specifically targeted. I want a mutation here, 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 and I want 50, 100, thousands of them. I mean, people, they can do thousands of these things. So it lets you dig through a genome and find mutations for whatever phenotype you're looking at. So here's another one that became a tool that is so powerful now, it's incredible. So the Cas9 protein, the protein that Alisa was talking about, that both the speakers before me were talking about, it has a catalytic function. It actually cuts the DNA. You can actually eliminate that function so that the protein is a dud. It doesn't cut, okay? So it's a dead protein. So now what you can do is you can take your little piece of nucleic acid, you can take your protein, and you can bring it onto a very specific region of the genome, but not cut. So you're thinking, well, what's the purpose of that? And that's what I'm thinking anyways. So here's one example. In your genome, you have genes that have to be turned on, turned off. Your genome controls that through your development, through your response to environmental stimuli. So now you can do this artificially. So the picture that's shown there is your wonderful Cas protein that's dead, that doesn't cut anything. You can put a little bit of another protein next to it, an effector protein or an activator, and you put your little piece of nucleic acid at the same time. Now you're going to bring that effector protein at a very precise location on the genome. You turn on activation, you activate expression of that gene. So you can artificially turn on and off a gene at will in your genome. That's incredible. So you can say, what happens if before that you were killing the gene, killing its function. Now you're saying, what if I turn it on? What if I turn it off? On, off, on, off. What if I turn these 2, these 3, these 4, right? Because you have multiple guides. So now you can go combinations and permutations of turning things on and off. So you can toy with the expression of your genome and just not knock it out and see what happens. And you can do that again in a very, very precise way. So here's another example. Actually, that's the same example, isn't it? Activation domain. It's the same example, yes. So the effector system is a little bit different. So in one case, you turn on the expression. In the other case, you turn off the expression. And there's variations on the theme on how to do this. This one I find really interesting. So Alisa showed you before that you could use a green fluorescent protein, and it makes things turn green. Right? It's just, we do that in biology all the time. You turn green, you turn red. So in this case, all you have to do is you take that wonderful Cas protein, again, that's non-catalytic. You attach a fluorescent protein onto it, and you take your little guide again, and you put that in there. So what's going to happen is it's going to bring your protein to a very, very specific location on your genome, and it's going to turn it green right there. Right? And in the picture that's shown up there, you can use a red fluorescent protein, a blue fluorescent protein, a yellow fluorescent protein. So you can illuminate very precise regions of your genome. You turn it on, and you can ask the question, where physically is that location of that genome during my cell division, during expression of you can ask the question, can I get access to that part of the genome? Because our genomes are tied up in little balls around proteins. Sometimes that controls gene expression. Can I get to it? Can I get to it? Can I turn it on? Can I turn it off? So some of these tools they're pretty creative. This is the latest one that came out, which I thought was even more interesting. You can actually So far I've shown you Cas9 is on, it's active, it can cut the protein, or it can't cut. So in this case here, you can have a system where you can conditionally get it to cut or not cut. So you can put the protein in there and you say, I want it to cut now, don't cut, don't cut. So the way you do this is you take the catalytic active protein, the Cas9, you split it in two. This part doesn't work, that part doesn't work. When the two parts come together, it works, okay? So they've used this in optogenetics, or so you can actually shine a blue light on your organism, and when you shine the blue light, the two pieces come together and they cut. So it's a very powerful tool. Now you choose when this thing cut, not the organism. So you can do the same thing. They actually used small molecules to bring the two pieces together. So these are very, very powerful tools to ask questions and answer hypotheses in terms of how genes work and how genes function and the dynamics of your genomes and everything else. So I just wanted to give it a short examples because I was asked to do this of what we use some of these tools for. So my entire lab, I'm turning into a CRISPR lab. It's incredible. Everyone's using it for different reasons. This is just one example of a project for a PhD student who I think is still in the room somewhere. Damien's back there. So one of the things we want to do is we want to replace the old ways of generating energy and chemicals and all sorts of things. So I'm showing the old way up here, and this is our new way of doing things. We want to be using renewable substrates to produce either fuels or chemicals. It's not an easy, simple process. I'm just put this picture up there. It's a little bit complicated, but if you want to turn a tree into a fuel, the way you kind of do this is from the top down, you take the tree, you crunch it up, you put some enzymes, and at the end you end up with something that looks like this thing here, which is really nasty, but it's a hydrolysate full of sugars. And then you ferment that to whatever you want. So the problem that everybody encounters is this thing here, which is your substrate, is very, very toxic. Animation. We all like animation. It's really toxic because of all of these different things. I'm not going to go into the details of this. So one of the projects that we had in the lab is we were asking the question, can we engineer or evolve a strain of a microorganism, yeast in this case, so it'll be more resistant to this really toxic stuff? So we actually did this. We started with a weak strain of yeast. We mutated it, and it was resistant to all this junk. We sequenced the genomes, or the genome of the thing that was resistant. We identified the mutations, but then we asked the question, what are these mutations doing? Are they really responsible for the phenotype or the resistance that we're looking at or not, and what are they contributing to it? So then we busted out a wonderful CRISPR tool, and we started bringing all of the mutations one at a time back into a weak cell, and then we asked the question, are we turning this weak cell into a resistant cell, or are we turning it to a weak cell? And if you turn it to a resistant cell, you got the mutation you want, and if you don't have the mutation you want. So the tool basically allows us to explore a lot of mutations and a lot of genomic questions pretty rapidly. So this is one example that we use, and I think that's my last slide. Am I under 8 minutes? You didn't wave, so. Okay.
Thank you, Vince. Always illuminating, always good. And the second thing I want to ask, if you, just for you guys to remember, other than Vince, is the on-off dance that he did. If you saw, I don't know if you noticed you were doing it, Vince. That was pretty good. So I'm going to hand it over now to our next speaker from the Political Science Department, who's an associate professor, Dr. Francesca Scala, whose research focuses on Canadian and comparative public policy with an emphasis on science and health policy. Did you want to come up, or did you want to speak from there? Perfect. That's fine with me. Yes.
I don't have a PowerPoint presentation.
Perfect. We'll mix it up.
I didn't understand.
You can't dance. Dance it down.
Just to be sure. But yes, so I'm an associate professor of political science, and my area of specialization is public policy, health, and science and technology policy. So as a political scientist, I see the discussion on CRISPR, I like the term, through the lens of politics or power and conflict. Right, and politics is really viewed as understanding the competition of values. The two questions. One of the questions that we ask as public policy scholars especially in the area of science and technology policy, is what is the role of the state, what is the role of government in regulating, in governing this area of research. And in Canada, we have a very long history in policymaking in the area of reproductive technologies and genetic engineering. Many of you know that we had a Royal Commission in 1989. And 3 years later, we had a final report. And it took about 12, 13 years before the federal government actually initiated, developed, and passed legislation. And it was a real struggle to come together and to provide some kind of a position that reflected a variety of societal views, the needs of the scientific community, as well as recognizing what the federal government could actually do with the tools it had available to it. So when we look cross-nationally, we see that governments have put in place different regulatory systems. So Canada is more often than not regarded as adopting the middle of the road approach to regulating reproductive technologies and human biotechnology. While the UK, as was presented earlier, we can see that just recently they announced that they got the green light, or researchers got the green light to edit an embryo, the human embryo. So. And then we have, Germany, where it has put in place several bans on some of these technologies. So cross-nationally, we see that governments have responded in a different way. Whether that's a good thing or bad thing, that's us for society to debate. But the other thing that we should also look at is what is the role of the ordinary citizen in deliberating on these types of issues. So more often than not, when we look at research on science and technology policy, ordinary citizens, societal groups, segments of civil society are typically marginalized or put aside when deliberating on these policy issues. And some say that's legitimate, right? Citizens don't have the competencies, they don't have the knowledge, they don't have the technical skills or knowledge to effectively deliberate, contribute to a policy discussion. Others argue that given the social and political and ethical implications of these technologies and these technological advancements, that it's necessary for government, for as well the scientific community, to reach out and educate the public about these issues, and also to provide forums in which citizens can actually possibly contest, challenge the knowledge claims of the scientific community. So in many countries, what have been put in place are different deliberative mechanisms. So we see in Denmark and in other Scandinavian countries, we have consensus conferences on nanotechnology. We have citizen juries on prenatal diagnostic techniques and so on. And this is a way of bringing in citizens and empowering them to exercise their own scientific citizenship. And in my view, that's extremely important. We do have, sometimes these debates are framed as a scientific and technological debate, and oftentimes they're framed as economic issues as well, because countries are striving to create favorable environment to encourage this type of biotechnology these advancements, because there are economic benefits to do that. But how does the government and how does society reconcile that need for scientific progress and for reaping the economic benefits of that with claims being made by civil society that these are potentially detrimental to their communities? So I'm thinking about some of the issues that come up is the notion of designer babies. Are we going to use CRISPR to somehow genetically modify out or delete certain medical or health conditions, right? So for some, this is a very good thing, and for specific diseases like muscular dystrophy, cystic fibrosis, and so on, and even the patient advocacy groups representing these diseases are very much in favor of them, and they're actively involved in this type of research. But there's also the other side, and the other side, what do we say as a society when we have parents who would want to use this technique to produce a child that reflects themselves? So here I'm thinking about disability advocates who argue that I want a deaf child. So I want to make sure that I will have a deaf child, and I will want to alter the genetic code in order to do that. Or, and there have been legal cases where deaf parents have gone to court to use prenatal diagnostic techniques to make sure that their offsprings will carry that gene. So these are some of the issues that come up when we're talking about it. So the benefits are there, and they're valid, and I think the economic benefits and even the benefits for curing, many diseases is there. What it will do for healthcare costs, some people are advocating for this because, given the current state of healthcare, given the escalating healthcare costs and so on, this could be, potentially good. On the other side, in terms of insurance, if we have this kind of data, if we can do something before we actually have a condition, and if we don't, what does this say of whether or not, we have access to insurance, whether we're going to be denied insurance, and all those kinds of challenges. So as a policy scholar, I advise, or I recommend, that we think about, and even maybe at Concordia, we think about having these kinds of consensus conferences, bringing ordinary citizens in so they can actually come in, deliberate on issues, they can be educated on the issues, and they can also sort of speak to experts in a deliberative forum. And another recommendation that I would make is having sort of this exercise here, this cross-disciplinarity approach to studying CRISPR and other kinds of technological advancements. When we bring in a diversity of experts to deliberate on the issue, to come up with some kind of consensus, to an approach that will reconcile, again, the needs of the scientific community and address the concerns of civil society. It's a challenge, but mechanisms are there, and I think from a policy perspective, I think we're well placed to do that here at Concordia or any university setting. So that's it.
Thank you very much. Is it still on? I'm on. Thank you very much, Francesca. That was awesome. I want to just, my pleasure to welcome our next speaker, Assistant Professor Matthew Barker from the Philosophy Department. Are you coming up?
Yep.
All right, come on up, and let's go on to the next one. Thank you, Matt.
Okay, so I want to cut straight to the ethical concern. Oh, is it okay? Did I cut out? Okay. I want to cut straight to the ethical concern that I think is raised most frequently about CRISPR in the media, but also frequently by scientists themselves. This is this idea that basic research, seemingly very innocent research of the largely academic kind, using CRISPR with human cells in particular, is going to be a slippery slope to designer babies. And if you look for articles commenting on CRISPR use, you'll see the term designer babies coming up all the time. And the worry is about designer babies by germline modification, right? So these are permanent edits to human genomes that last from that point forward through generations. So there are many questions we can ask about this. Here's one. Is there really such a slope, let alone a slippery one, right? This is not really rhetorical. It's an open question. And to show that there's not an obvious answer to it, let me just put it a little more provocatively. Does basic research using CRISPR, removed from any clinical setting, raise the probability of designer babies any more than learning algebra raises the probability that you'll become a millionaire, right? So that's not to express my own viewpoint on how to answer this question. It's just to say there's not an obvious answer to it. And to answer this kind of question, we need to undertake a lot of very careful scientific investigation, empirical investigation, because it's largely an empirical question about whether doing this kind of basic research is going to raise the probabilities of this thing that is constantly expressed as a concern, designer babies. I'm going to put that aside. I want to focus instead on this related question. Even if it turns out to be a slippery slope, would designer babies by germline modification really be ethically bad? That's certainly more of a philosophical question than the previous one. So I want to focus on that. So I think the most common way of answering this question often helps itself to a very simplistic view, so I've called it the simplistic dichotomy view. This is the view that divides all genes into one of two types, either genes that help in some way to cause disease or disability, or all other genes, those that don't, okay? And then it is thought to be pretty simple to draw the ethical line. You draw it roughly like this. It's ethically permissible to modify genes on the left, right? Disease and disability genes, and not those on the right. Now, perhaps as the name suggests, I think this is far too simplistic a view. And I think a better view to take up is a spectrum view. This makes it clear that what matters is well-being, and in particular expected well-being when we're talking about modifying human embryos. Right? So on this view, you can plot genes with respect to how they influence expected well-being. So genes that fall far to the left on the spectrum are ones that will influence it most negatively. So diseases that are often described as catastrophic diseases will turn out far to the left on this spectrum. Diseases like Duchenne muscular dystrophy, which is being researched using CRISPR right now at Toronto Sick Kids, Canavan disease. But then moving up towards the positive side, but you'll see I put a 1 in the middle there, that's neutral. If a gene falls right in the middle, it's neutral with respect to expected well-being. So we're still on the left, we're still on the negative side. Disabilities like being deaf are often thought to be negative, right? So they're often plotted there as well. Things like having an IQ of 80, which is sometimes actually called having a disability, also thought to fall to that side. And even things like being female are thought to, much less than the other traits, of course, but they're thought to negatively influence expected well-being if, for example, you're born female rather than male in certain northern communities in India. Okay? And if you vary the community, it might turn out that being male is the one that slightly negatively influences expected well-being. The point, though, is that this shows how simplistic the dichotomous view up above was, right? There's no such thing as a disease or disability gene simpliciter. There are some genes that have more effect on expected well-being, some that have less. So now the question becomes, where do you draw the ethical line once you've realized it's more of a spectrum, right? So some quite conservative views about modification, and these include some, certainly not all, religious views, will draw it way out to the left, which is to say, well, no modification is permissible. It's all prohibited. Right? Even modification of catastrophic disease genes. Then a far more liberal position, this is one that basically puts the line right in the middle and says it's ethically permissible, perhaps even required when possible, to change genes from those that would negatively influence expected well-being to those that wouldn't, right? That would positively influence it. So that kind of view has been given a pretty rigorous voice recently in the bioethics literature with something called the principle of procreative beneficence. So I just use PB for short. Here's what it roughly says. If you're a prospective parent who is creating a child, then there's a strong ethical constraint binding on you to create the child with the best chance of the best life. Okay? There are arguments given for this principle. I don't want to look at the arguments, but rather what this might mean for CRISPR if you just take this principle off the shelf. Okay? So you might be able to draw this implication for CRISPR use from procreative beneficence. It says if you are creating a child and germline modification by CRISPR is an option thought to be safe, so obviously we're already talking into the future now, at a future time, then there is a strong ethical constraint binding on you to use this option to remove any disease or disability or other expected drag on child well-being. So that's a kind of procreative beneficence implication for using CRISPR. I think this kind of view has several problems, and I'm just going to highlight a few of them for you. One stems from a kind of hubris that I think a lot of us have about disability. So although many severe diseases greatly reduce well-being, to the surprise of a lot of people, the best empirical research suggests that most disabilities do not greatly diminish expected well-being, or well-being if if you've got the disability for that matter. So instead of putting the so-called disabilities where I did, it turns out once you look at the empirical literature that things like being deaf or having an IQ of 80 are far closer to the middle on the spectrum. And in fact, most disabilities reduce well-being by only a small amount, if at all. And once you realize that, I think you'll see it's not enough to warrant germline modification in a great many cases. Here's another problem, this one stemming from intellectual hubris. It's that even if we reach a stage where all evidence, I mean, this is fanciful now, right? But imagine we reach this stage where all the evidence we have, and we have a lot, suggests germline modification by CRISPR is safe for humans. Even if we reach that point, we might still be mistaken, right? Now, if we reach that stage where we have this massive evidence that all points towards safety, then we couldn't rely on the evidence to see the mistake. I've just told you we're in a state where all the evidence points to safety, right? So the evidence is not going to tell you that you've made a mistake. It's suggesting safety. Well, once you're in that state, how is it that you're going to assess the possibility that you might be mistaken? It's very difficult to figure out how you assess the possibility of mistake in those kinds of cases. That's when humility is called for, especially when you're dealing with magnitudes as large as you get with germline modification. Another problem with this is stems from unjust inequalities. So following the procreative beneficence ethical constraint would greatly raise the chance that many marginalized groups, including those with disabilities or oppressed visual minorities, would face even more inequality and injustice than they already do. So suppose you agree, okay, I'm not a big fan of this procreative beneficence principle as applied to CRISPR. There are other liberal pro-modification principles you could pick up instead. This one I'll call the principle of parental autonomy. And in a sense, well, I'll tell you what it says first. It roughly says, if an adequately informed parent has some capacity to influence her child's future, as she autonomously chooses, then no other person should manipulate that capacity of hers. Right? This puts a lot of stock in parental autonomy. So in this kind of case, it's sort of like saying, let's not put a line anywhere for all people. Let's leave it up to the autonomous choice of parents. Now, I think you can actually find the same kind of problems I already mentioned for the procreative beneficence principle for this one. But I think this one also faces a different kind of problem. It's a rather extreme conceptual problem if it's correct, if I'm correct about it. It's basically that the principle can be self-refuting. Okay, so here's how that goes. Suppose Anna is a parent. She believes that this principle of procreative autonomy is true and manipulates her child's germline via CRISPR. If she does that, she also seems to imply that the principle is false. Okay? How does that work? I'm suggesting to you that in this kind of case, where she professes to believe the principle, it uses CRISPR to manipulate the child's germline, then that action might also imply that she believes the principle is false. That would be to say it suggests she's irrational, right? Here's why. In doing that, Anna would count as a person who is manipulating the child's own capacity to later influence her own kids, his or her own kids, right? So if she exercises her autonomy to modify the germline of her child, there's a sense in which she's stripping the autonomy of her child to do the same thing at a later date. To some degree, of course, the child might be able to say, well, I don't like what happened to me by CRISPR, so I'll use CRISPR again to reverse things, right? But that's still having had a limitation put on the child, okay? And more specifically, it's to introduce a kind of irrationality in the principle. I think that's it. Yeah.
Thank you, Matt. Thank you very much. I'm going to introduce our last speaker, and then we're open to a little bit of a discussion between the panel and for questions, so you can start thinking about the things that might be brewing now. I'm going to introduce David Morris, who's also from the philosophy department and is also the department chair, to come on up and give our final presentation before we open things up. Yeah, I think they'll, sure, and they'll probably put something up if they can. Or either way, David.
Sorry, I need to start my timer. Okay, philosophers are something like the CSI ideas team. We sift through things that people write and say for clues as to the concepts behind their thinking. We then do a sort of forensic analysis of these concepts via our knowledge of the workings of these concepts and the history behind them. We do that to glean insights and help improve our thinking. Curious remarks or results can put our team on the scent. When I first started reading about CRISPR, what stood out, especially in the popular press, was a general sentiment, explicit or implicit, that CRISPR, the way it edits, regulates, etc., is or could be much less worrisome than prior genetic technologies. Why? Overtly, this has to do with at least two things that are linked. First, the specificity, that is the far higher and already growing precision of CRISPR. Second, the way that specificity lets CRISPR do important work by editing an organism's own genome in quite precise ways. But why should that relieve worry? I suspect that concepts of agency are deep in the background here. CRISPR can deftly alter an organism to let it better realize kinds of activity already indicated in its genome or activate potentials already belonging to it, whereas with prior genetic technology, this is more difficult, less feasible, less precise, more prone to error. And in the press, the prior technology sometimes comes across as blasting an organism with external genetic material that alters the way the organism would act on its own. To give an exaggerated example, prior technology can appear as the alien inserting its eggs into an organism to make it do alien things, whereas CRISPR is like a medication that can restore or bolster an organism's own activity or activate previously mute parts of its own developmental repertoire. So CRISPR would thus be less invasive, less pernicious, more natural. My purpose, though, isn't judging CRISPR on the basis of that observation itself, which is anyways too simple, and CRISPR can also insert alien material and so on. Rather, what I want to note is how deeply concepts of agency and activity and passivity shape that kind of assessment of these issues. And then I want to raise some conceptual considerations that complicate matters. Now, the activity-passivity distinction, which I'm now going to discuss for a little bit, it was key to ancient Greek thinking about differences in gender, sexual orientations, and social and political roles. Roughly, activity, manliness, citizenship, and freedom were linked together and contrasted and valued over passivity, womanliness, homeboundedness, and submissiveness. That's important because when Aristotle invented biology, that conceptual matrix prejudiced his results, especially his view that females supply eggs as passive, malleable material, whereas male sperm activates eggs via a form that specifies what an organism is to be. Simone de Beauvoir showed how related prejudices about activity versus passivity proliferate in sexist thinking. And philosophers and scientists have shown how these prejudices can and do persist in and can distort contemporary biology, for example, in certain concepts of sexual selection. These issues are also in the background of resistance that we find in the history of science to crucial insights that happen to be proposed by women biologists such as Barbara McClintock, Lynn Margulis, Jane Goodall. And whatever position one takes on the recent controversy regarding Eric Lander's article, Heroes of CRISPR in Nature, you can't detach it from this kind of background. The discussion around it shows it. I want to move away from that, though, because what I'm trying to do is illustrate how conceptual forensics can alert you to issues about how you conceptualize things and how that can distort science, results, discussion, etc. But my example actually is of special significance because I really don't think that we can do biology without concepts of activity, passivity, agency. These are key to grasping life or organisms and entities of that sort in the first place. Yet, I think the history of science shows that we tend to draw our understanding of key terms and concepts in the history of science from human life and our presumptions, only to find that the living phenomena we're studying challenge our conceptual models. For example, our experience of our own agency can lead to a classic liberal individualist kind of view of agency as a sort of already given inner power, already prepared to choose from within itself. But that concept of agency and activity can be very misleading as a basis for judging the good or bad in genetic technology. Indeed, biological phenomena belie efforts to conceptualize the genome as a sort of already fixed inner agent that can decide things. As scientists know, in its actual operation, genetic material is intensely dynamic, networked, recursive, modulated by epigenetic factors. It's less like a single score specifying a piece of music to be played, and more like a group of jazz improvisers playing changes while somebody shuffles, rearranges, and covers or uncovers the sheet music. And the development and function of multicellular animals, such as ourselves, is in many ways polygenomic, since bacteria and their incredibly fluid genomes are factors as well. So we shouldn't fall into the tempting but too simple thought that CRISPR is a game changer because it crisply edits an organism's own score. CRISPR intervenes in much more complex dynamic phenomena, and the genetic agency of an organism isn't clear-cut, but spreads and blurs over space and time. Finally, a more challenging point. The genetic dynamism of organisms lets them develop not simply according to a fixed genetic plan, but in deeply plastic ways. And that's key to evolution. While CRISPR may allow us to change the way organisms work by deft edits to their own genetic material, when we do this, we are, as it were, not letting organisms play out some of their own future plasticity. We're channeling them to a future we're selecting. So we need to be thinking about our edits not simply as intruding or not on the current or past genetic agency of organisms, but intervening into a future agency that could not yet be fully defined, because it's also environmentally sensitive and opened in ways that we can't fully predict. Indeed, my thought here is that if agency is to be agency, it has to be open to its own future as not yet fully determinate, a future to which agency is passive. Here we run into conceptual and ethical problems about the future that interestingly echo the ethics of climate change as involving responsibility to future generations, and there was something in Matt's talk that was sort of like this. So we need to also remember that things like our judgments about the good of antibiotics went badly wrong because our presumptions about the genetic agency of bacteria were badly wrong. We missed how genetically promiscuous and plastic they are. The issue here that I'm thinking about is rather the plasticity of organisms as an in principle, not fully predictable resource of potential future benefit for all. Might our edits modify or curtail plasticity itself or lock possibilities out of play? My conceptual and ethical point is that our responsibility is not simply assessing whether proposed edits now lead to ethically acceptable or desirable results. We must assess their impact, especially in the case of proposed germline edits, on organismic agency that has a plastic potential that we can never fully assess, since such agency is inherently passive and open to its own future. So there's a deeper tension here between our science and technology as working toward a predicted future and the future as not yet being fully determinate. Biology, I think, is wondrous because it plunges us into the thick of life and evolution as generating endless forms most beautiful. And it thus gives us a deep encounter with beings who make themselves in surprising ways that challenge our presumptions. And they thus give us an encounter with a time that is not of our own making. And it's that time, I contend, to which life holds us responsible.
Thank you, David. Thank you very much. And thank you to all of our panelists for a great round of talks that hopefully opened up some ideas and some thinking here. And based on typical kind of how we're going to go forward, I think from here we might open it up to a little discussion between the panelists before asking for questions. And when questions come, I believe we're going to use the mic that's in the middle there, and we can bring people to go up there. I just want to get a sense from the group, how many questions do we have on our mind so far? Can people raise their hand if they want to ask a few things? So 2, 4, 3 more coming up. There's a couple. All right. So hopefully, as we go forward, what I want to challenge the panelists with at the moment is generating a few more questions or ideas that might spark some ideas in the crowd as we go forward. Because I know there's more hidden. I can see so many eyes. So I want to just open it up for a quick kind of go through, very fast response from each of the panelists, if you guys are okay with it, to what you heard from the other panelists. Alisa, do you want to just start quickly? And this is just off the cuff, your first thoughts, and as quick as possible in that kind of a sense.
So one thing that I guess jumped to my mind, and this was as I kept hearing this designer baby thing, I'd like to emphasize that when we talk about editing human embryos at this point, we're talking about a human embryo that's only kept for up until the 250 cell stage. These are not embryos that will be born into people. So it's very carefully controlled, and I think scientists, I'd like to think, have very strong ethics in place, and we understand the implications of what we're doing. So I hope that you can give us some faith in that. And the scientists that have been approved for this work are really working on very specific diseases, and these are diseases that would fall into that far spectrum that Dr. Barker was talking about. So these are very debilitating diseases, and this could be the only hope for some of these people. And I'd like you to think about going into the past and like that discovery of antibiotics, vaccinations. There's many things that have really changed the way we live, have improved our lives, but at one point were considered very unethical, right? So I'll just
Thank you.
change
Francesca.
from there.
I'm going to mix this up a little. We're in a line of scientists on the one side and other ones. We should have mixed it up. Francesca, quick thoughts. Brief is good. Just going to pop it out. Yeah.
You know, I think most citizens have a lot of faith in science, and I think that's a good thing. However, you know, some critics or sociologists of scientific knowledge would argue that we could look to the past and see how the trust in science perhaps was misplaced. Right, so I think if there is a forum where citizens and non-scientific experts and researchers come together to deliberate on these issues, I think we can sort of increase trust in science. But I get a little nervous when scientists say, trust us, and we'll do the right thing, because there have been past instances or examples where that hasn't happened. So we can think about, you know, hormonal treatment for women, where it was supposed to be a good thing, it was supposed to be healthy for you and so on, and then we see, well, it leads to breast cancer, right? You know, the whole debate on vaccination is an interesting one, the HPV vaccine. I know.
You'll get your turn.
I know, you know, but I do think there's nothing wrong with citizens or non-scientists challenging, you know, the validity of scientific claims. I mean, that's a good thing, and I think the scientific community should welcome that. Right, so that would be my only comment. Yes.
Thank you, Adithi.
So I want to add.
Vince,
to that.
please.
So,
I'm going to open it up to you guys to jump in. Was that you or me? Which one of us is magnetic?
So,
It's Vince.
Magneto.
Go ahead, Vince, please.
So I actually agree with the political scientists more than anything else. Part of the problem I've noticed with CRISPR and other technologies that are coming on board, and this is where the ethics get a little flexible with scientists sometimes, is nowadays the bigger the splash and the crazier thing you do as a scientist, the more chances you have of getting the front page of the big Science and Nature magazine, and the more chances you are of landing large NIH funding and everything else. So I've been sitting back and watching some of these scientists, and some of them are doing stupid things, and they're doing stupid things just to demonstrate how powerful they are, how powerful the technology is, and they need to be pulled back in. So I'm with you on that one. I would not rely for 1 minute on the ethics of some of these guys at Harvard and Stanford are on tremendous amount of pressure to make the first page of the journals. So I think you have to consider that. I don't know if any of you guys read this thing about gene drives. You didn't follow? You need to read those papers. I'm going to toss that out there, and I'm going to see how things react. So there's a technology called gene drives that was published where, so we're talking about germline modification. This is even worse. So you can engineer CRISPR into an organism so what they're looking at is mosquitoes. So they want to solve Zika and malaria and everything else. So they put this into a mosquito, and they put the mosquito into the wild. The mosquito, every mosquito that this mosquito mates with becomes infertile. So basically you can put a poison pill into the mosquito population and kill all mosquitoes, all of them.
Over time, right?
Over time,
Yeah.
right. So this is pretty powerful, right? It's self-sustaining. The other thing that comes about is something called genetic pollution. I don't know if you ever heard that term. I think that's a fantastic term. Genetic pollution is, are you starting to put things in places you shouldn't put things in, and then not knowing what the consequences are going to be. But this gene drive thing is, and it's actually been demonstrated at the lab scale. You can actually do this. I hate mosquitoes. Good thing, right? But think about the consequences of that technology beyond mosquitoes, and it's very scary. It's a self-propagation of your genes through engineering.
All right. Vince, you're generating questions. I can hear it already.
Oh, yeah.
At least me, the journalist in me, wants to know about some of who these scientists are. They're doing things you don't trust. Well, we'll come back to that later. Matt, what do you think in terms of this?
Let's see. Well, first of all, I'm glad that Alisa made the clarification about germline modification being very different from the basic research that's going on. The question that I addressed is this worry that kind of basic research that seems very cordoned off from things like germline modification with humans is nonetheless a slippery slope, right? That's this worry that keeps coming up, and I think it hasn't been assessed very well. It's a bit of a scare tactic. But it's still nonetheless worthwhile to think about what the ethics of that are once we're able to do that kind of thing. So if you want to think about more near-term ethical issues, one thing I guess I would like to throw out, just out of interest for scientists in the room, is in what way do you think ethical constraints arise in work with CRISPR at this point now, just doing basic research, not in connection with human germline modification? What kind of ethical constraints do you see arising just in basic research? And I guess I would encourage people to think about that in a different way than is usually portrayed in the media. It's terrible that often in the media you get this kind of opposition between science and ethics is seen as this kind of obstacle that the scientists are warned they have to be conscious of and jump over. But I think a lot of the time, in very basic research, when you're in the heart of some scientific project where you feel most removed from ethical considerations, they're still there, but in the opposite kind of way that people would expect. Instead of there being some kind of constraint on you, often ethics are actually a motivation for the thing you're doing. So for example, when Galileo invented wholly new kinds of observations to try and learn about the nature of the solar system, some people, like his colleague and friend, supposedly Caesar Cremonini, refused to look through the microscope, right? And to still continue to use the old type of observation for testing the same hypotheses they were both interested in, right? That seems like a failure of ethics, right? And you might think that the same kind of thing is going to happen with CRISPR. I'm sure it already is. So CRISPR is allowing very powerful new ways of making observations to test hypotheses. And if you have a grad student, for example, and you're ethically concerned about their welfare and their career and getting a job, are you doing something wrong if you say, look, use this old technique, you get the old observations, when you know there's a telescope now? Right?
Interesting. Let's go to an undergraduate student, if you don't mind, to respond to the initial question and just your thoughts in general about basic research in general. Are there ethical concerns already, a positive, negative, restraining, forceful, and those kind of ideas?
Yeah, so I think that last point that was brought up is a really good one about, I guess, like being not forced, but not trying new techniques and using old ones. It's a very good point, because as you come in as an undergraduate student, you should be trying new techniques even though you don't want to, or maybe just to open your eyes up. So I guess when there's like questions like that, it's very interesting. And I know there are people like that I've heard, and even my lab, that don't want to use the new technique and they want to use the old one. So I guess questions come up in that sense, and yeah. So,
David, what do you? Any comments or things to add to get us going? I have a few questions to ask, but I'd love you guys to keep going if you want to.
Well, one thing is that I think we all have to be alert to the ways that the very thin slice of this that we get in the popular press doesn't really give us a glimpse of the real practical implications, the real possibilities for discovery. And Vince, the presentation really was nice about the way this lets us pose new questions. But there I also think it's sort of striking to me that to do the science of living beings well, we have to learn how they work from the organisms, and also to do the ethics of living beings well, we have to learn how they work so that we don't make what I'm talking about as conceptual mistakes. And CRISPR itself, the thing that we're worried about, it seems to me like it gives us striking insights into how things unfold with genetic activity on a very fine-grained spatial and timescale in ways that we can't have done before. And it also allows people to very rapidly go through cycles of questioning where I think the difference isn't just quantitative, it's qualitative, because you can do things across the time scale of a research team in a lab. So it's striking how at once this technology that we're worried about also gives us resources. But I think the thing that I'm most interested in is really being alert to the ways that we human beings, I think, ironically, our evolution badly equips us with the kinds of conceptual structures and perceptual tendencies to really get how these complicated, distributed, evolutionary, ecological processes go. Like, we're just not good at thinking this kind of stuff, and we have to learn from the organisms. And just one thing that also is striking to me, at least the way it comes across sometimes, is the thought that most people didn't really, like, even though there were results, people didn't notice the striking thing that bacteria have a kind of version of the immune system, right? So this thing that we failed to know about bacteria is now being deployed to do all these things. And I think there's a sign of a kind of conceptual humility that I think is part of what Matt was talking about, that like we have so much to learn, I think.
Okay, see some nods. You want to jump in, Alisa?
Well, I guess I just wanted to go back again to bring up the ethics of using CRISPR in general. And actually, it's Jennifer Doudna, who is, of course, one of the main players in helping to move CRISPR from this sort of basic technology to something we're all using in the lab today. She felt that the field was just moving way too quickly. And she has made several comments at TED Talks. She's really been very vocal about how we still don't fully grasp the concept of what this technology can do. And there is something that we call off-target effects, where it could be that we think we're only targeting one part of the genome, but we could be hitting other parts of the genome too. So we really need to be careful to know that this is being done properly before we move forward. And she has actually been one of the advocates to say, hey, let's look at the ethics of what we're doing and let's slow down a bit.
So Vince, I often hear in technologies like this that slow down is often what comes up quickly. Do you think this is a time to slow down, or is this a time is this one more example of a scientific technology that is a point to, some people talking about the same old issues about, not saying that they're bad, same old issues about gene editing and predicting the futures, how things will go? What's your feeling on the actual speed that we're moving at with CRISPR?
So I think the genie's out of the bottle, but that's two different questions, I think. So germline editing is one. This is the view I view it, and I think about it, and it goes from humans and as well as animals. I guess we are animals, but so I think tread carefully there. But everything else, every other aspect. One of the things I think it's important to grasp is we can in the lab do science so fast using CRISPR, so precise, it's so fast that we can speed up discoveries, but the application at the discovery doesn't have to be CRISPR related. I can find out a lot about cancer genomics, I can find out a lot about diabetes, I can find out a lot about using this technology, and once I have that knowledge, the solution doesn't have to be germline editing. So I'd say speed up CRISPR research as much as you can, but not necessarily germline editing. That's the point. the speed at which we can do things and the scale and the magnitude is just through the roof. Don't slow that down.
So am I understanding correctly that you're thinking that we're spreading, breaking out the basic research on getting CRISPR better and using it, and the clinical and/or germ headline research over here is the thing we need to take time with and think a little bit more.
Make sure you keep those things separate.
Francesca, thoughts?
No, I agree. I think that's a good. I could get behind that. Definitely. But what's interesting, and again, I'm not an expert in the field, but I remember discussions during the Royal Commission. When societal actors were saying we're really concerned that the slippery slope argument, that this is going to happen and that's going to happen, and the scientific community would come back. It's like, we don't have the technology to even do that, so put that off the table. Right? So that's the way they sort of stopped the debate, because there's no possibility of us doing that. And I remember distinctly that there was talk about the 3-parent baby that happened in the UK, and they were talking about is the possibility of having the genetic material of 3 different parents and so on, and they said, well, that's impossible. That's in the future. That's we don't have the technology, and it happened. Right, in the UK. Now, whether or not it's a 3-parent baby, and we have to sort of deconstruct that and so on. So I think there is a slippery slope argument, and they're compelling and so on. But I don't think we should dismiss them outright. I think we sort of have to investigate them, examine them, and really sort of advance a more sort of not rational necessarily, but a more deliberative, educated discussion on them. Yeah.
Fair enough. Matt, on the slippery slope. I know you're in there. I saw the great graphic. I was sliding right off it.
Yeah, I guess one thing I'm not sure any of us has touched on yet that I think is very relevant to the slippery slope concerns. I mean, I tried to address them saying they're not as obviously of concern or as obviously not of concern as people think. I mean, they're tough. You got to think about whether these are really legitimate concerns. But one thing that makes them very relevant and worth thinking about is the regulatory environments being fractured. And those are the environments in which these kind of questions are going to arise. So, I mean, in plainer terms, the regulations and governance in one country are very different from in another country, right? And so even though you might say, well, in Canada, look, we've got a prohibition on human germline modification, in others there isn't, and science isn't contained to any one country. So it takes a lot of very careful social science work, actually, scientific empirical work, to figure out exactly what the slippery slope concerns are, and it's really tough because there's such a fractured regulatory environment. Yeah.
All right. I'm going to ask people to begin to think about their questions and get ready to ask a few with the microphone as we go around. I'm going to just see if there's any final comment from the panel or if we're ready for questions. Vince, yeah, please.
Maybe one thing we didn't address. I like to toss.
Yeah, please toss.
There, so.
Actually, Vince, can I ask everyone to toss out one final thing while Vince is talking?
Okay.
Because I think that's a good way for us to kick off.
I'll be the first toss.
Yeah, toss.
So Alisa kind of brought it right at the end of the talk. Kind of mentioned it. But one of the things that's really, talking about slippery slope, that's really dangerous that's going on right now is all the intellectual property battles that are going on. So both Berkeley and Harvard have filed for patents, or the Broad Institute, and so there were patents even on synthetic genes. And so there's patents that are coming out there that are so powerful. And the question that I keep wondering is, do you really want to give one company, one university, the control of that technology? And the number of patents have just exploded, but this is going to be the longest, most expensive, most difficult patent battle you're ever going to see in your life. And these are just the patents that we're aware of. There's so many other patents that have been filed that we still don't know about. So I'm just putting it out there just to, that's my final bomb.
And we're talking like predictions of $1 billion technology type thing.
And then some more.
Right.
Oh yeah.
Melissa?
Actually, I'll just echo that because one of the, and by the way, that 150 patents that I showed you, and that was back in 2014, so that's not current. And over 60 of those was coming from MIT, and over 30 of those from one person. So it's not even do we want control from one company, but do we want control to one person? So I'll just echo Vince to throw that out there.
So I guess I guess what I can add to this is that from an undergraduate perspective, I think the lack of like communication, I think as scientists, we find it hard to communicate how much we know or how little we know because it's so technical. Like as an undergrad, I know that's what I struggle with a lot.
You did great. That was awesome. No, you did.
But I feel like a lot of these issues with the public and the questions that come up, I think a lot of this stems from the fact that there's a lack of communication or an inability to communicate properly what we know and what we don't know. So I think as an undergrad, maybe that's where we should start, even like in schools and in classes, just like improve that first, and then we know we can get where we want to.
Francesca, anything to throw out?
Sure. I guess going back to my issue about engaging citizens in decision-making processes dealing with science and technology. How do we do that? Should we do that as well? I teach a course currently on citizen engagement and public policy, thinking it would inspire my students. And more often than not, they're like, let the experts take care of it, right? Which is like, because the idea that, especially,
yeah. Like, let's do it. I got this under control.
So there is, there is a lot of, there's a privileged status that experts enjoy in our society, especially scientists, and should we provide forums, discursive and institutional forums for non-experts to come in and help deliberate and decide on these issues.
Great. Matt?
I guess one thing that would be interesting to think about is how best to make regulatory bodies more diverse and representative of the population. So in the past, I've worked for an ethics review board at a medical school at a university in the States. And I was a philosopher, I guess, but I was working for the board. I wasn't on the board. So they snuck some diversity in there in one way. But I was alarmed to see how large a proportion of the people on the board were physicians with no training in ethics whatsoever. There were some exceptions, but it seems we need some kind of regulatory body for regulatory bodies, right? Like we've got to find better ways to form these things. It's not, it's about engaging citizens of various perspectives, but even just engaging experts of varying perspectives. And of course, there's lots of appetite for that in a room like this and among panelists like this. But somehow it's not always showing up in practice, and I just wonder how to fix that.
Awesome. David, final thoughts, and then we'll move to open questions. We will have quite a bit of time, so feel free to throw your questions in, and we'll go from there.
So two clusters of thoughts. One of them is thinking about Socrates, talking about how he knows that he doesn't know, and thinking about the ways that our technologies for investigating the world, our instruments, they make certain things available to us, but they can also hide and conceal things from us and get us not to notice. So like our fMRI machines make certain things available, but they might cloud certain things about the time scale of observation and so on. So, I think we need to be alert to that. And this, I'm like a biology geek, I think, in some kind of way. Like I try and read as much about it, and I love biology because of what it shows us. But I think even as CRISPR is giving us this new possibility for research, we should be alert to the things that might get skewed away, or just be prepared to be surprised, I guess. And then what Vincent said about ownership, I mean, that is striking. Right? So each of the technologies and sciences that we invent, especially nowadays, tends to open up new domains of ownership, right? So now a lot of the things that we own, we are not allowed to repair because they have software inside them that's owned and copyrighted. And, so our technology just gives us new sites where people can own stuff. And here I wanted to cite the philosopher Michel Foucault with his concept of biopower, which is basically the thought that our biology, the things that we do that help us with our health, sets up new domains where we get to be controlled in various ways by the people who in some way control those technologies and medicines. So, your access to medical care, who gets to have birth control, et cetera, et cetera. And even as we're researching and learning more, I think I'm suspicious, just given what human beings are in politics. We're like, some people are going to grab for power. So whether we like it or not, this research that we do, all the research that we do in any domain, like, we live in a world where people are grabbing for power. All right. And now they're grabbing deep into the roots of things that have been working for millions of years to be their own thing. So, how's that for a learn?
There we go. Thank you very much to the panel. I'm going to open it up to questions. You can just raise your hand, and there's a microphone. Is any questions from the crowd? I'm just going to start right here. I got you right there. There we go.
I have a question for Francesca. Actually, I'm a biology professor, lecturer. I teach genetics and genomics, and also I teach intro biology to the poli-science students as well. So I feel like I'm sort of myself interdisciplinary and so on. And a lot of things that I heard today, and I'm familiar with the CRISPR, but the controversy is similar to what we had in this the pre-implantation diagnosis that you can select and choose the embryos. And basically, a lot of things, those embryo manipulations, we can actually, you don't have to do it. We just select and choose already. We already have a designer baby systems. So I think that guidelines still probably apply. That's one. But another, so I haven't actually asked the question. Sorry, I have a question.
Oh yeah, that's supposed to be instructed.
I'm supposed to be.
Questions should go right to the point.
Yeah, sorry. I have a question. Yes, question is, who decides on the science policy? Because to me, the science policy always falls behind. And the good reason is that the example is the genetic testing. And you are probably fully aware that currently they're trying to introduce the bill to not to misuse the genetic information. But this is like after over 20 years of genetic testing, and why are we deciding now? Why did we not have this discussion and the law already placed? So because you talked about, having the consumer or whatever, the public and a part of the policy, but this is like non-policy. So I'm really not sure where is the leadership and policy, where is the will and who decides and the science policy. And I would love to know if you know, or, we would love to get involved, so.
Thank you.
That's a very good question.
So why?
Why is this? Well, it's this notion of regulatory science, right? So, people who study science and technology in a lot of, policy areas, more often than not, we rely on epistemic communities, we rely on policy communities, and we rely on experts, right? And when it comes to regulatory science, government science or, the science that government relies upon is usually not up to date. And that's the problem with making policy in this area. We have advancements that are coming, forward and so on, and our legislation, the tools we have in place to actually address them, they're not effective anymore. Like, do we use our laws, our criminal sanctions, right, to do something about it? Well, the scientific community is going to be up in arms. But under the Constitution, the federal government has only that option, right, to come in. We have, a bureaucratic system where, things have to be vetted, where we have to, see whether or not it goes against the Charter of Rights and Freedom. So, and politics comes into play. Right? So many times we have delays because, the officials in the bureaucracy are talking to the scientific community or talking to CIHR, and they're saying, don't pass this, or, there's a lot of lobbying that happens behind the scene. So there's a lot of different reasons why a policy can't keep up, because policymakers don't have the expertise. They have to rely on the experts that they want to regulate, right? So there's an issue of information and power there as well. I don't know. I'm not sure what the answer is. I mean, we also have a federal system where there's provincial system, there's universities that are involved, there's private institutes. I mean, there's so many things happening. So that in itself is an interesting question for policy scholars to look at in terms of delay. And another question that you raised as well about, we're already doing prenatal or pre-genetic testing, in terms of discarding, picking and choosing those embryos and so on. And this, I would think, I'm thinking about sort of the anti-abortion groups, where they would see this possibly as a positive thing, because you don't discard the embryos that are deemed defective. You go in and you fix it, and then you can transfer and it can implant in the uterus. So I'm just thinking that this could actually, not shift the alliances, but could sort of prove an avenue for, those who have a moral objection to embryo research or prenatal diagnosis. That could be sort of a way forward to sort of address some of the issues of surplus embryos and destroying the embryos that are deemed to be, suboptimal. In that sense.
Yeah, Vince, please, and then we're going to go to the next question.
I just want to toss a comment. This is in the old days. So I spent a little bit of my life working at Environment Canada. We used to do research to help policies and regulation, and the government would come to the scientists and ask the questions: should I regulate based on this? Based on this? We don't know. Let's do the science. Provide the data. Let's regulate based on. We have the data now. So what's happened in government funding has been a horrible effect on that. So there are certain things that you should not and cannot rely on industry to do. You need to protect your citizens, and you have to have some sort of social conscience. And this is where the government needs to step in and do the research to generate the data and the information that will control those regulations. So that research almost is non-existent in government level anymore. So that's a problem. I would say, and I don't know if Graham is still back here, but I would say, and he's
No, he's hiding. He's got his hand up.
He's slowly dragging me into this, but even Rémi Quérillon, the chief scientist in Quebec, says, as scientists, we need to engage the policymakers more, mostly to fill the void that's been there.
All right, I'll let that hang. I'll go to our next question. Thank you. Then I'm going to go, when you're done, you pass it in just behind you there. Okay, thank you.
Hi, I'm a biology student. I currently work at Neuroscience Lab. We work with genetically modified organisms. And this is important for the question because on the subject of public engagement, I work with a lot of people who don't have a scientific background. They are psychologists. They're BA psychologists. They don't really know much about biology or physiology or anything. So when I tell them that I work with genetically modified organisms, it's a touchy subject because I can't really explain to them what they are. They just shut down. I say that they're modified organisms, and they, needless to say, I've ended friendships because of that. Not on my side, but the question is, how do you have a conversation with people who don't want to listen to arguments?
I love this. I love this one. No, Vince is not going to answer because he doesn't listen. All right. No one's going to. They don't listen. This is a wonderful question. Panel, give us some insights. Give us some answers if you have them.
I'll just start, but I think this is really for you guys over there.
No, I think this is for everyone.
But, well, honestly, I've had this discussion with a lot of people, and I remind them that since man has cultivated, or women have cultivated and grown crops, we have been genetically modifying organisms. We've been doing it by selection over time. We're picking the biggest cows, the best corn, and so this is something that we have been doing since we've had farming, really. So I would use that as an example for them and say, like, we have just taken it to, yes, another step where we're maybe bypassing this need to screen through thousands of crops to find the one corn that's growing better. Now we're just modifying it and having that process occur much faster. So that's kind of how I would present it, but
comments?
I'll let you guys take over.
So what was the exact wording again? What do you do with people who won't listen to arguments? Is that it?
It's really complicated.
Mm-hmm.
To actually have a conversation, because when you start using terms that are not common language,
Mm-hmm.
that are not for the layman.
Yeah.
When you start talking about the lab, and when you start talking about using, say, genetic tools, say, artificial selection, a term that is alien to certain people,
Yeah.
how do you have that argument?
Well, you have to use as few terms like that as you can. I mean, don't use jargon. I mean, and part of education, I think, is really not becoming skilled at that until you're much farther along. I mean, when I started graduate school, I didn't know how to do that. I didn't understand my own subject matter enough to switch from communicating in one way to in a more accessible way. So it just takes time. But to respond to the general question, what do you do with people who don't listen to arguments? My approach, and my students are probably tired of hearing this because I harp on it in every philosophy class I teach, is to take a charitable approach, right? And instead of starting with the view that my beliefs are different than this person and they're not even going to listen to my views, ask why they hold their views. Try and understand what the strongest position is behind their views. And sometimes they might not have a strong one, but by asking those kinds of questions and inviting an open discussion rather than trying to force your opposing view on them, you'll come to see what better arguments might underlie their positions that they might not even be aware of. So it's an intellectual charity is the way I think to handle that.
Please, Francesca.
Yeah, and I also think, in some of the research that I do, this whole notion of lay expertise, right? That we have lay people who over time develop expertise and are able to communicate with, citizens or certain segments of society. I have a colleague who went to, who does research on AIDS, and he went to a conference, and there was an AIDS activist there who had become a lay expert on the virus and on the disease and medicine and so on. And what he did was that he put up, a journal article, and he deconstructed it. Every line that had a lot of jargon and terms, he simplified it. And he said, this means blood. This means an injection. This means that, and so on. So we sort of have to try to, to make that, to use accessible language. So everyone can participate, and the playing field is level in that sense. So I think this notion of lay expertise is extremely crucial. So you may not be an expert in the content and processes of science, but you can certainly understand, what is being communicated, and you can certainly contribute to advancing that type of expertise. So I think that's crucial as well. It's about the language that you're using, and again, going into the process being open. Not having an agenda and not having an argument that you want to advance, but really, it's sort of a Habermasian type of approach where, you create a forum where everyone is deemed to be equal and not trying to advance any kind of agenda in that sense.
But you need to speak about that too.
I don't think I'm supposed to. I could say something.
No, say something. Science journalist, because I think sometimes it's how you frame it as well. The perfect example, so the word GMO triggers emotions like you wouldn't believe. So I'd be careful of this.
Yeah.
I'd be careful of using the word GMO. I gave an interview with, what's her name, Takahashi at CTV, and she, right before the camera turned on, she leaned over and said, "Don't use the word GMO." And the cameras went on. I went, so how you frame it, because you're going to get completely different outcomes. So I'm going to let Dave pick it up between something like synthetic biology and GMO.
I won't say much, but we should talk in that kind of sense. I want to go to things, but what I will say is that the idea that underlying many of what people are saying is this idea of diversity of views, that even in this room, whether or not we're all scientists that think this, or all biologists, all philosophers that think this, or people who go there or that way, that even there's going to be a huge diversity of views. And respect for those diversity of views and understanding them, underlying the arguments where each one's come from, is the place that you need to get to be able to get there. Framing issues are going to heavily weight how you can access those diversity of views, how things are approached and talked about will close doors when they shouldn't be closed. So they're very important things that the panel is saying. But I don't want to talk too much because I could talk forever. And I want to go to the next question.
So some of you up there might know me. I'm a grad student in philosophy. Matt's my supervisor, as it turns out. And I thought of directing my question specifically to Matt, but I'd also be interested in if any of the other panelists want to weigh in. I wanted to ask about possible shifts in our social attitudes about disability with the kinds of avenues for research that we're opening up here, and specifically also about the possible erosion of disabled communities and how that affects the kind of expected well-being, as Matt put it, of disabled persons. So just really quick, the example that makes me think about this is there's a great blog, Impact Ethics, and they had a couple of people that went to the International Summit in December on genome editing. And there was a great post from someone a deaf person, who was writing about how this summit was not planned in a way that was accessible for deaf people. And they made that very excellent point, I thought, that something like CRISPR actually bears the possibility of eroding these kind of flourishing language communities that are needed for instance, for a deaf person to actually to flourish, to achieve an ideal of their own well-being. So this seems to put us in a difficult situation where for deaf persons or deaf families or members in deaf communities who choose not to have any kind of treatment, they are actually put in a worse position by those that do, because the community that they require to actually fully flourish is being shrunk. So I was just wondering if I could get your thoughts on these issues.
Matt.
Yeah, well, I think the underlying thing that needs to change is just our outlook on disability. I mean, I think we're just factually mistaken in lots of ways about disability and about the well-being that many disabled people enjoy, which does not at all imply that they don't face social injustices, right? That's a very different thing. That the fact is that many disabled people have much higher well-being. I mean, there's data on this, right? So most people with disabilities report much higher well-being than those without disabilities presume they'll report. But that's not to say they don't face social injustices. That, of course, they do, and it's remarkable that they can achieve the well-being they do while facing those injustices. But so this issue about the deaf community, this is a good example, has come up in the literature. I've got a paper under review about this kind of topic right now, and one of the reviewers said, it's a little strange to defend the idea that you shouldn't modify in order to delete deafness from the population for the sake of deaf people who already exist. It's like saying, too bad whatever person is going to grow out of this embryo, you're going to have to grow up to be deaf because other people who already exist need that. And basically the reviewer was saying, like, that's a strange burden to put on that young embryo, right? So that's a different kind of perspective to look at. I actually think there's a problem with that kind of response. When you're selecting, for example, by pre-implantation genetic diagnosis between embryos, the embryo, if it has a genetically determined predisposition towards deafness, and suppose it's also very likely going to lead a life of reasonably high quality, by allowing it to persist, you're not harming it in any way, right? The alternative is you select against it in order to help people think they're helping that future person, right? But if you select against it, it won't grow into a person. It'll go in the garbage can. So as long as it's going to grow up with some reasonably high level of well-being, then I don't think you're harming that embryo by allowing it to persist, growing into a deaf child that won't erode these kinds of communities.
We are nearing the end. I'm getting the signal from the big boss person to do it up. Kim, you can ask the final question. Yeah, it's going to have to be the final question. But in the final question, I'm going to ask just to quickly, if the panel could pick up on the question for a final take-home, soundbite journalist on video kind of thing to go on. Kim.
I just had a really simple question about the relationship between media not being out there, but actually infiltrating, in a sense, the practice of science and being integral because of the rise of a kind of celebrity culture, not just for pop stars, but in the scientific community that actually influences practice in a way that may be detrimental. So we're talking about the detrimental effects of the technologies of CRISPR, but in effect, what we're also talking about are some of the problems within our culture and the way that media operate. That also has an impact on science. So I guess I just want to flip it a bit.
So, final comments, the role of media, of science celebrities, and of a lot of the things that have gone on. If we can ask each of the panelists to say their final words, we'll move from there. I know there's always more questions and more things to ask, but you'll have a chance to catch the panelists at the end. And I want to make sure Andrea has a chance to speak and we haven't stolen all your time.
So just quickly, that's a really great point, because I know that I, as a scientist, suffer with this idea that now we have to use the most sexy techniques and we have to compete with these scientists that are getting all of the attention for the money and the funds to do the research. And a lot of us, we can't publish in the same way unless we do these, so there's a lot of pressure. And we may be running, again, maybe a bit ahead of ourselves. And I personally struggle with this a lot, actually. And I do think that by creating these celebrity scientists, it is hurting the scientific community and as a whole, because these people do tend to get most of the money. They get the top papers, maybe not always reviewed in the right way, right? And maybe their grants aren't always reviewed in the right way. And it's at the detriment of the rest of the community. So,
yeah, I think it's in the same sense when you're like working, like as an undergrad trying to choose a project, when you only hear about what's in the media. I think you miss out on so many cool things that you can learn. And I feel like that's a really, really, really like disadvantage of media and what they focus on. Like if everything was focused on CRISPR and making germline mutations, I don't think I would be in the project that I'm in right now. So it makes a difference. Yeah.
Vince.
So that's a good thing, right? I don't know. I've had the experience of speaking to a lot of journalists in the last year, and I have a mixed feelings. And sometimes I feel like the way media is going now with the short little snaps of information is really more detrimental than the way journalism, good science journalism, used to be. So this is more of the picking point for me. But and they won't let me review their articles. So they write it, and I tell them, send it to me, I'll fix it. And they refuse to do that. Because it's against their ethics.
No, I agree with Vince. It's hard. I have to say, there isn't much out there in the mainstream media or in the newspaper on CRISPR, right? So I had to go searching. I had to go and do my little Google searches and so on. But and someone had to mention CRISPR, when I was invited here, they're like, do you want to say something about CRISPR? And I'm like, what? What's that? And I had to do my own investigation. So it's not, I think, science journalism, I'm not an expert in the field, but, they're not doing the watchdog function that they should be doing. And I think that's very detrimental to the communication of science to the public and the public understanding of science. That's extremely problematic for me.
And when you do the watchdog, it's sensationalism.
Yeah.
They go,
yeah.
Which
it's either Frankenstein or rockstar celebrity
which doesn't
scientist,
help the cause,
right?
yeah.
Matt.
Well, there's so much I could say.
Remember, I asked for a soundbite. The good
soundbite.
Yeah, lay it on me. Something I can put on, a scarf.
A return to more public funding than private funding, I guess. Or maybe that's not the right way to put it. It's not maybe private funding per se that's the problem, but to have companies recognize the benefits for them of more public regulation. I think you've heard views up here about wanting to keep the basic research separate from the more applied concerns that people have. So there's a will there, but I think it's going to be very hard for those things to be kept apart if the right kinds of regulations aren't brought in.
Thank you. David, take us home. Nice and quick. I'm already getting the looks that I've
Philosophers
taken
don't
too
do
long.
sound bites. We chew indigestible things for thousands of years.
That's all your time, and we're...
So do I get to say something or no?
Yeah, you do.
Okay. You journalists are good at so like science is a human discipline and we're evolved beings, and one of the things that's striking to me is the ways of thinking and reasoning with one another that have evolved to do well. are now in a terrible mismatch with the time cycle of our media and communication apparatus. And it's interesting to hear, scientists feeling like they're being trapped in those kinds of nets. And one thing that's been striking to me about CRISPR is the timescale of the phenomenon, right? Like, I know it's been going on for a while in science, but from the outside, it's like since last summer or something like that, all of a sudden, it's this thing that's changed the world. And that, I think the timescale of that transformation is perhaps a kind of novelty for biology itself, and that needs to be thought about. And then there's also just something interesting too about, like, I think we're thinking about media and timescales and how things move and how there's something very different about the rate at which science does its research and precipitates its research versus, say, the way we're slower in philosophy. And it would be interesting, I think, for us at Concordia to talk just someday about the timescale of our research and what that means for our research lives. Because you lead, like, you're different animals than us over here. And it's good to be in the,
I finally got my soundbite.
around.
I got my soundbite right there with the different animals. David, thank you very much. The panel, thank you very much. I'm going to give them a big round of applause. Really well, really well. Thank you guys for all your insights. I'm going to ask Andre to come up and give us our closing remarks.
Thank you, David. Well, ladies and gentlemen, that is Beyond Disciplines. And David, Matt, Francesca, Adithi, Vince, and Alisa. What an awesome panel. Yeah. It was just awesome. And Dave as a moderator. One thing about it, and I kept repeating this last year when I visited all of our 27 units, I spoke about keeping this enjoyment of what we do, the fun of this intellectual endeavor that we're on, and the sharing of it, the conversations about that. And this illustration of tonight is exactly what Beyond Disciplines in the Faculty of Arts and Science and Concordia is those conversations that actually create insight, create vision, understanding, thought-provoking, that you come out of this room and you think, whoa, I never thought of that. And those conversations can happen here. They can happen in this sort of free kind of. So you see in front of you a proud dean of Arts and Science, Concordia. So there are a few other people that I'd like to thank that I've worked really hard behind the scenes. And this idea of Beyond Disciplines has come up as a team effort. And three people that actually worked quite a lot for this first event, and I'd like to mention them. It's Kim Sawchuk, our Associate Dean, Research and Graduate Studies, and I believe she has some students working on a podcast, because this will be podcast.
Not immediately, though.
No, but it will be podcast. James Camlett, Associate Dean, Faculty Affairs, and Paul Joyce, Associate Dean, Curriculum. They all contributed to sort of make this sort of first CRISPR thing to come alive. So thanks to all of them. Big thank you to Liz Farr, who's our communications advisor. And to Maria Soil and Deborah from my office that actually contributed to make this event a great success. So now I want to thank all of you for coming all the way to the Loyola tonight. Not many of you have left, so I gather I interpret this as very meaningful. But there will be a next one. And the next one will be about telling stories. It's in March. And telling stories is part of what we do in scholarship, in teaching, and everything else. And there again, we will have a panel that will sort of span across the faculty. And you're all invited, and I have all your names. So I'll check. Because you all signed up to come here, so I have all your names. So better be there, and we'll have a bigger room, so we'll have more people come. So thanks, everyone, for coming. It's great that you took the time to do this. Now, we have a tour of labs. Vince, I'm not sure how many people signed up, but we have guides who will take you there. And then you'll join the reception afterwards. And those who are not on the tour of labs, there's a reception right now. So,
Vince want to say something?
yeah.
So there's people from my lab who are giving the tours, I don't know. Oh, back there, waving their hands.
They're there.
You can actually CRISPR something in the lab if you want to.
So thanks to our guides as well. So thank you very much. Great.